Today I'm going to finish talking about aspects of humoral cellular immune responses, as well as talk about some aspects of cellular innate immune responses. And I just realized I set up one thing wrong here. Or maybe I didn't. Okay, sure. So we talked about a number of antimicrobial peptides in general last time as one part of the humoral innate immune response, but then we moved into thinking about complements. And this is the view of complement that is in your textbook. This is a view of complement from a different textbook, but they both kind of are giving us the same general idea that there are three different ways you can start the complement cascade. Sometimes we refer to those three as initiation pathways. No matter which initiation pathway you start with, you end up at this same middle area. And once you get to that middle area, then we have ways that we can get rid of the microbe, which are known as the effector functions. And so you can see that again, here's your textbook's version, here's the other textbook's version. So you can see the initiation. Here they're calling it termination. I usually just go with effector. And you can see there's also this sort of common ground in the middle. And what we did on Friday was we talked about initiation via the classical pathway. So we did sort of this one branch and got to this middle section. I also have listed that information up on the board. So remember that with the classical pathway, we start with antibody binding to the microbe. C1 then binds to the antibody. Oh my gosh, C1, once it binds, is an active protease. It can cleave the next member, C4. C4, once cleaved, is an active protease. It can cleave the next thing, C2. Yes, all of these, when there's cleaving happening, we get little fragments. We don't really care about the little fragments, so I didn't tell you a lot about them. But once C2 is an active protease, it cleaves C3. Specifically, it cleaves C3 into C3a and C3b. C3a we're going to actually need to care about eventually, so I actually wrote it here. C3b, oh my gosh, active protease! Cleaves C5 into C5a and C5b. And you can see sort of the parts that I've listed as up here in the classical pathway initiation. You can see sort of this part I put in a box that is this central part. And once we have our C3 and our C5 cleaved, we're at that middle place. That's really getting to C3 and C5 is the goal of all of the initiation pathways. And now we can get rid of our microbes. And so what I want to start with now is how we get rid of the microbes from our C3 and our C5. I realized I drew this a little low here, so I might put them up here and give myself a lot of room. And then what we'll do is we'll circle back and talk about the other initiation pathways, the other two ways you could get to C3 and C5. So we want to get rid of microbes. We don't want to just cut up proteins and do nothing exciting. So here is where we kind of left off. And as I told you last time, there are 4 different ways that we can deal with microbes, though one of them is going to get like one sentence and the others will get a bunch more. The most famous way that we deal with microbes is through something called the membrane attack complex, or the MAC. This is also showing you the sort of details in terms of individual proteins, and as well as the actual downstream process. The MAC is really coming from C5b and its activity and it having been activated. And when C5b is active, it turns on 679. You don't need to know the specific really details of that, just know it's the rest of the numbers, 6789, are involved in the MAC. You can actually see that happening up here from your textbook, C6, C7, C8. C9 is the kind of cool one, because C9 basically makes a pore in the wall of the microbe. And so once you get 5, 6, 7, 8, 9, you basically make a hole in the microbe. You put a whole bunch of copies of C9 together and you make a pore in the surface of the microbe. And this is actually pictures under an electron microscope of those pores that are made in the surface of a microbe. Here you can actually see an end-on view of one of those little tubes that's in the surface of the cell. And so this is the membrane attack complex. And basically, it just is poking holes in membranes of the microbe and lysing all those microbes. And so you get the microbes lysed because you attacked their membrane. Not super complicated. Pretty obvious how that kills microbes. And so the idea is that as we've been attaching all these proteins to the surface of our microbe, it's almost like we're labeling it and saying, proteins, come here and destroy. And then they're going to come and destroy with the membrane attack complex. So this one's pretty straightforward. It is definitely the one that people usually remember the most. This isn't the only way that we can get rid of microbes using complement, however. There's another thing that happens, and this is happening not so much from C5b, like I showed you, we saw the membrane attack complex. This one is happening from these little fragments, C3a and C5a. I didn't care about any of the little fragments before, but with C3a and C5a, they actually also have an effect. They can act on blood vessels. They have receptors, and so you can see here C3a and C5a acting on a receptor. And that receptor could be on a macrophage or a basophil or an eosinophil or a neutrophil or a mast cell. And that can induce inflammatory responses. And so we can get changes in the blood vessel wall. So this is sort of the blood vessel before C3a and C5a. And you can notice that the blood vessel gets leaky because now extra proteins can leave the blood and try to go into tissues to try to get rid of things. It also makes it easier for cells to leave the blood vessel to try to go to that area and get rid of microbes. So you can think about C3a and C5a as proteins that are sort of helping to make the environment better for recruiting other things by inducing this process of inflammation. Sometimes people refer to C3a and C5a as the anaphylatoxins, though that's a bit sort of outdated terminology, but it is listed there. So we're going to get rid of microbes by lysing. We're going to get rid of microbes by basically making this nice environment where we can get other cells and other proteins to come in. And we're going to do one additional thing. Really two things, but we'll deal with it. And this includes a vocabulary word that I have to tell you about. Still, a lot of years later, I snicker with this vocabulary word. Some of you guys who had micro with me have heard me tell this story. But this vocabulary word is called opsonization, and we're going to see opsonization come up again later in the semester as well. When I learned opsonization, I learned it as like my professor gave us a little like story to help us remember, like an analogy. And when I got to my first ever immunology exam, I could only remember the analogy, and I couldn't remember actually the definition of it. And so I wrote a really funny answer for my definition of opsonization. So I'm going to tell you the analogy, but don't write that as your answer eventually. So the idea with opsonization is that you stick something to the surface of a microbe. Here you can see C3b is stuck to the surface of a microbe. You can sometimes we also say you are coating a microbe with something in opsonization, and that makes it more likely for the microbe to get phagocytosed. So opsonization is basically adding some kind of coating to a microbe to make that microbe more likely to be phagocytosed or to enhance that microbe's phagocytosis. What my professor told me back in the day was that opsonization was like butter. It's like something you put all over things to make it tastier and more likely to get eaten. And so if you look at my first ever immunology exam where I had to write the definition of opsonization, I wrote butter. So I recommend not writing butter. But the idea is that you're coating with stuff. In this case, you're coating with complement. We're going to coat with something else later in the semester. But in either case, you're coating with something to make it more likely for that to get phagocytosed. So C3b can opsonize. It can coat the microbe. And you can see that there are some receptors, some complement receptors on the surface of this phagocytic cell. That complement can bind to those complement receptors, and now we can phagocytose that microbe, eat it up, get rid of it. And so that's our third way of getting rid of microbes because we started the complement cascade, is we've now made those microbes more likely to get phagocytosed. One thing that you can notice about opsonization, as I show it to you here, is that opsonization requires a complement receptor. It requires that phagocytic cell has a receptor for complement that is bound to cells. I mention that because in truth, there are tons of complement receptors and they can do a bunch of things. I'm not going to go through all of the details of all the complement receptors and all the things they do. But what I also want you to know is that one other thing that can happen, and it's related to a complement receptor, and it's often coming via C3b, is activation of adaptive immune cells. And so everything we're talking about with complement is part of the innate immune system, but one of the things that can happen when we get complement on a microbe is we can be basically saying, hey, adaptive friends, please wake up and start doing something too. And we can start to kind of turn on other parts of the immune system. And so we've got a bunch of complement receptors. Many of them are on adaptive immune cells. You can see B cells and T cells listed in a few places here. And so complement being deposited on a microbe can also help to activate the adaptive immune response. And I'm not going to go into any more details about that until much later when we actually, talk about the adaptive immune response. So that's the whole bottom part of complement. So we talked about the classical pathway, and the ways that we get to C3b, and C5b, getting activated, which is that middle area, and the ways that we can get rid of our other pathogens. You can see here your textbook calls it perforation of pathogen membranes. That's the MAC. Opsonization and recruitment of inflammatory cells, which is happening by the inflammation happening from these guys. So inflammation or recruitment of inflammatory cells, like it says there. So that's sort of the detail of sort of the classical pathway and how we get rid of microbes with complement. This image from your textbook shows that the classical pathway is the last to be activated. And the reason why it is last to be activated is related to an awesome question that Brooke asked on Friday, which is that we want to remember that the classical pathway requires an antibody to start it. And an antibody is part of the adaptive immune system. So you can't even do all this till you already made an adaptive response. Lame. But we also have these other two ways we can start the complement cascade that do not require an adaptive response. And so they are likely to happen a little earlier. You can see that your textbook says that they're first or second to be activated. In addition, the classical pathway is really only found kind of in our jawed vertebrates. Not everybody can do classical pathway because not all organisms have the ability to make antibodies and have an adaptive immune response. But our other two pathways that we're going to talk about are present in many, many, many organisms, basically throughout all of the animal phyla and potentially in some other places as well. And so you can see that these other pathways, they were discovered after the classical pathway. Classical was discovered first, so that's why it's classical. The other ones probably evolved actually way earlier and were ones that were important first. And so we need to talk about those two. So one of the two pathways involves a protein called involves something called mannose-binding lectin. It's sometimes also known as the mannose-binding lectin or MBL pathway. Sometimes I call it the lectin pathway. In order to totally understand the lectin pathway, you need to know what the word lectin means. I'm going to tell you what the word lectin means. We're going to see another lectin way later in the semester, and I'm going to be like, okay, everyone, what does the word lectin mean? And you're just going to stare at me, but that's okay. So a lectin is a protein that binds to a carbohydrate. That's all it means, is it's a protein that can bind to a carbohydrate. The lectin in this case is called mannose-binding lectin, or MBL. So if you had to guess, what carbohydrate does MBL bind to? Mannose, exactly. So we've got mannose-binding lectin. From its name, we can guess it binds mannose. It turns out there's a lot of mannose on the surface of a lot of prokaryotic cells, not so much on the surface of eukaryotic cells. So you can see these little red hexagons are the mannose. And so the first thing that happens here is that microbes have mannose. You didn't really have to do anything there to make that happen. Now, a protein is going to bind to that mannose. And in fact, that is mannose-binding lectin is going to bind to the mannose. So, MBL, mannose-binding lectin, is going to bind to the mannose. If you remember the name of the pathway is the MBL pathway, it's pretty easy to remember this protein is MBL. You can see some images of MBL here. And if you look at MBL in these images, what do you notice about MBL? Yes, David. It looks a lot like C1, which is often described as an upside-down bouquet of flowers. Structurally, it looks almost exactly like C1. That's pretty much exactly what C1 looks like if you look back at the other things. What happened with C1 in the classical pathway? You can look at the board if you needed help. But yes, Jonah. And what did it do? Cleave C4, right? Became an active protease that cleaved C4. Guess what MBL does when it binds to mannose? The same thing. Oh, man, the hardest part of this is I keep losing my chalk. All right, there we go. We're done. There's the MBL pathway. MBL, as you can see here, here's MBL binding to mannose, looking just like C1, and it cleaves C4 and then cleaves C2 and does exactly the same things that C1 did. It just got to this location, not because there was an antibody there, but because there was mannose there. Otherwise, it's exactly the same. So now we didn't need an adaptive immune response. We just needed a microbe that had mannose on its surface. So you can see, pretty easy. And then we've got the third pathway. The third pathway is the alternative pathway. It is everyone's most and least favorite of these pathways. And you can see that your textbook lists the alternative pathway as the first to be activated. It is probably the first one to have evolved. It is the first of many times in this semester where I'm going to describe something and you're going to be like, that's stupid, and think that it does not seem like a really great idea. And you're going to learn that the immune system is incredibly inefficient. But there are some reasons why this is actually pretty important that we have this. So if you come up with, if you at some point during this description are like, I don't like this, this is a bad pathway, you're not the first to have had that thought. And in order to understand the alternative pathway, we have to think a little bit about the protein C3 itself. This is where we're getting into the chemistry, so I'm getting a little scared. You can see the C3 protein in extreme biochemical detail here. It's a blue blob. But what you can also notice is that we're showing you one bond in that protein. You can see we've got this sulfur bound to this carbonyl, which is known as a thioester. You can see that oftentimes we can have, when we cleave C3, it's sort of right at that thioester. But what's not really depicted here quite as well as I'd like it to be, and I will show it to you on the next slide as well, is that this thioester in C3 that you see right here is at a really weird angle. It's kind of like when I was at physical therapy last week and they were telling me all of my joints that were at weird angles. Yeah, it was bad. So the thioester is at a super weird angle, called a strained thioester. If you have a joint or that's at a really weird angle, and in fact under some stress at that weirdly weird angle for a long period of time, which is what is happening with this strained thioester, what could happen? Yeah, Jonah. It wants to not be strained. Yeah, it wants to not be strained. What's a good way for it to not be strained? Huh? Okay, what's a good way for it to not be at a weird angle? Don't overcomplicate. What? Break. A really good way to just get rid of all this stress is to break, right? And so sometimes C3 spontaneously breaks. And when it does that, it pretty much breaks into C3b and C3a. You can see this here as well. Here's my strained thioester. Sometimes it just spontaneously breaks. You can see this up here as well. Spontaneous cleavage of C3. Sometimes C3 is going to just spontaneous. Because this was named a bazillion years ago, it's known as the spontaneous tickover of complement. I don't know why we use those terms at all, but that's the term people came up with. And so we get this spontaneous breakage. When that spontaneous break happens, we've got, again, a bond that kind of wants to react. There are two options for what happens if that bond reacts. And which of the two options happens is based on what is the first compound this C3b comes in contact with. So it's sort of like whatever is the first thing it finds is the reaction it does. One option is that it finds water, which is at the top. If it finds water, it just makes kind of like a crappy C3b that doesn't do anything and no one cares. However, if it finds either an R group with a C double bond O, so an R group with a ROH, I guess, as you can see here, or if it finds an R group with an NH2, so if it finds either of those things first instead of water, then it reacts and attaches to that ROH or RNH2. Can anybody think of anything in your body that has an ROH or an RNH2 on it? Amino acids. So every protein in your whole body? Yeah? Every protein. Okay, cool. Anything else that has either of those things? Besides every protein? Thanks to the amino acids? I got at least 3 more. What? Nucleic acids? All the base pairs have OHs and NH2s in them? So every piece of DNA and RNA? So we got all proteins, all nucleic acids. What else? 2 more. Yeah. Cell surfaces, like specifically, I don't know, because of the proteins on the cell surface, or what else might be on the cell surface? What would you say? Lipids. All the lipids have this. So lipids are going to be on the cell surface. Alright, so we got protein, every nucleic acid, every lipid. There's one other thing. There's one other thing that goes with those. Every carbohydrate in your body also. So you can imagine there's a lot of this stuff. The probability of that C3 that spontaneously broke finding a protein, lipid, carbohydrate, or nucleic acid in your body, pretty high. And if that happens, the C3 gets attached. What that means is that in you, all the time, C3 is breaking. Break. All your C3s are breaking right now. And attaching to all your protein cells. You are getting C3 on cells constantly, spontaneously, through this process. If we think about this spontaneous takeover of complement. Here you're seeing the same thing. You know, it's either inactivated from going with water, or it will go straight onto that microbe surface because it's going to potentially go onto all of these things in a microbe. That's going to be super fast. Think about how fast, like, you didn't have to even do anything. The C3 already was spontaneously breaking. It could get C3 attached to it in the alternative pathway in like 1 second after getting into your body. That microbe's gonna get this C3 attached. And so if we want to think about something that's real fast to act against microbes, this is amazing in terms of being real fast to act against microbes. Once that spontaneous C3 breaks, and we get that spontaneous C3 binding to some surface, like the surface of a microbial cell, other proteins will come in. I'm not going to go through all the details of which protein in the order. You can see that there's one that's called factor B. There's one that's called factor D. But the basic way that we think about this is that for the alternative pathway, these other proteins, like factor B and factor D, help that C3 become an active C3 that can actually do the job of making more C5s. So basically, once you have C3 cleaved, once you have C3b, you get to make more C3b. And so often we think of the alternative pathway as just like a positive feedback loop of once you have some C3, you get more C3. And you can notice it has these other proteins, factor B, factor D, and propyridine. I mention those names literally because I want you to notice the names are like not C1, C2, C3 kind of names. So if you're trying to guess, if you're looking at a protein and are like, oh, I can't remember which pathway this is in, if it has a C, whatever, it's in the classical. And if it has some other wacky name, it's in the alternative. If I actually go through if I were to go through this in great detail, it would mean that I would get to tell you about an exciting protein. I'm not going to tell you about this, but I would get to tell you about an exciting protein called C3BBBBBC3B, which just rolls off the tongue. Yeah. We're not going to deal with that. But this is basically what the alternative pathway is. The alternative pathway has a couple things about it that are awesome and one thing about it that's terrible. One of the things about the alternative pathway that is awesome is the thing that I've already told you, which is its speed. The alternative pathway is happening basically spontaneously at all times in your body. So if we want to have something in terms of our innate immune response that is actually attacking microbes within seconds or minutes of that microbe getting into your body, this spontaneous chemical reaction is pretty good. And so the speed's awesome. The other really nice thing about the alternative pathway is that it can serve to amplify either of the other pathways. So it amplifies itself and it amplifies the other pathway. Basically, whenever you get a C3b, you also get to get alternative pathway going, and so you can amplify. And so I'm showing you this. You can imagine we might have one molecule of antibody binding to our microbe. And that's only going to allow us to have one sad C1 come and bind. And C1 could maybe turn on more than one C4, but it's not going to be a huge number. We're going to get, a little bit of all the proteins. But once you get to C3, every C3 can give you more C3s, in addition to giving you C5 and later parts of the pathway. And so instead of, even if you had one little sad antibody, once you get to C3, you're going to be putting so much C3 all over the surface of that microbe. And so this amplification piece of this is really nice. I showed you earlier with the membrane attack complex, all of these different pores on the surface of this microbe. That could have happened from one antibody binding. And just so much C3 getting deposited because the C3 is basically depositing in a positive feedback loop, which is not a positive loop. But there's also a problem with the alternative pathway. Jonah.
I was going to say, if C3 is constantly breaking down, how does it impact, I guess, the classical pathway with the C2 protease attacking C3, or does that even matter?
You make C3 in such excess, your liver, all of these proteins are coming from the liver as part of the acute phase response. You make so much it doesn't matter. Okay. Yes?
When you say it keeps making a bunch of C3, is it making the C3 like before it's cleaved?
It is. It's making the pre-cleaved C3, yes. Yes, David.
You said before that C3 can bind pretty much anything present in the body. How does it determine whether it should proceed because it's a microbe or if it's something that's itself?
Look at my next slide. So then, as I mentioned, there's also this problem with the alternative pathway. I showed you that here when we have this cleaved, this C3 that has cleaved, it can attach to ROH or RNH2, and we listed off a bunch of things that have ROHs or RNH2s. And so the question becomes, well then, how the heck how are we alive? How is it that we haven't lysed every single one of our cells in our body? Because we've all been around long enough that the C3 that's spontaneously cleaving should have probably managed to hit every cell of our body at this point in all of our years, and could have had a membrane attack complex and this inflammation and all these other things. So we should have poked holes in all our cells. We shouldn't even be here. It's kind of what David's asking. How is it that we're not constantly destroying all of our cells? Because with what I've just told you, we might constantly destroy all of our cells. And the way that you asked the question made it sound like you're like, oh, I bet there's a way that it knows, like, not to have a chemical reaction on our cells. Nope. The way that it actually works is that our cells all express complement inhibitors. And so all day long, your cells are getting covered with complement, and all day long, your cells are clearing the complement off. And so every single step of the complement cascade has an inhibitor that some cells express. And so there are cells that like stop C3, or there are proteins that stop C3 from becoming active and that actually turn C3 off, or that disrupt other parts of this process. Some that actually get in the middle of the pore if the pore tries to form on your cells and blocks the pore. And so basically 24/7, the alternative pathway is trying to destroy your cells. And 24/7, your cells are just like, go away, stop, go away, and inhibiting complement. And so as I said, this is not super efficient. You can imagine this is, kind of wasteful, but it's really useful in being so fast at protecting you. Because the microbes don't have those inhibitors in many cases, and so they're not going to be able to get rid of the complement. So they're going to be getting coated immediately, they're going to be potentially acted on, while your cells are going to stay safe. And so in fact, it's, even though this is super wasteful, you're wasting all of these C3 proteins all the time, it's actually evolutionarily better because of how it's just going to be saving you and especially helping you deal with how fast those microbes reproduce and just keeping the numbers down. Because remember, we talked about replication rate and the fact that they can replicate just so fast. So that is the answer. So whenever you are sitting around thinking you're doing nothing, know that you are in fact inhibiting complement, making sure it doesn't kill you. So thanks, complement inhibitors. So sometimes all of these preformed proteins that act super immediately, sometimes they do the job. They get rid of the pathogen. We're no longer infected, and we live happily ever after. sometimes these preformed proteins aren't good enough to get rid of the microbe. Some of this is because microbes have evolved sneaky ways around them. Some of this is just because microbes are tricky. And so now we actually have to have the next part of the innate immune response start to act. And this is really going to involve cells of the innate immune response instead of preformed proteins. So now we're going to actually get into the cells that are going to be acting in the innate immune system. You can see that this is going to take a little bit of time. Those cells have to sort of get revved up and get ready to go. There's another term that you've likely heard many times that's really the term for what we're talking about here in terms of the cells of the innate immune system acting. And that term is inflammation. Inflammation, I'm really going to use as sort of this broad term for action of innate immune cells. People often hear the word inflammation and they think, oh, that's bad. I don't want that. But I want you to realize that inflammation actually has a super important physiological purpose. Inflammation is really important for defense against infection, as well as things like tissue repair and response to homeostasis. Inflammation can go wrong and cause some problems. We'll get to that later. So it can go wrong, but if you didn't have inflammation, that'd be bad news bears. Inflammation is actually super important for you. When we see the inflammatory response happening, we are going to be seeing a lot of activity of two different types of cells. One of those cells is the neutrophil, your friend from last week. The other cell is a macrophage or monocyte, also your friend from last week. So these are two different innate immune cells, two different myeloid cells that we saw last time. Both of them, as are listed here, do phagocytosis, but they have some differences in actions. And so today and on Friday, we're going to see some of those differences. We might only really get into the macrophage today. We might deal with the neutrophil more on Friday. And so we're going to kind of talk through the process of what's going to happen in this very classic inflammatory response. This is your textbook's figure. What I want you to notice in this figure is here we've got a capillary. So we're just going to say here's our blood vessel. You can see there's blood cells in the blood vessel. Under the blood vessel, there's some other kinds of tissue. There's also some tissue up here. And this is showing you we've got your skin. So this is your skin. This is kind of the area of tissue of the skin on top of the vessel. And I don't know why this is, but I swear, like almost every immunology class, at some point, the professor has given this exact example, and it's always been, and then if I slammed my hand very hard on the podium and gave myself a cut in my skin, and I'm like, why are we slamming our hands then? But yes, the idea is that you got some kind of cut in your skin. If you really want to slam your hand on a podium, that's fine. But you could imagine other ways you got a cut in your skin as well. The moral of what's going to happen is you're going to have some kind of microbe, like this bacteria, that's going to get into your skin through this surface wound. And we need to see kind of the steps of exactly what is going to happen. What I want you to notice in the before part of this is that there are some immune cells hanging out in tissues. Here, this is listed as an effector cell. I'm going to give it a different name in a second. But the idea is that we got some immune cell that's already there hanging out. And that immune cell that was already there hanging out is going to be the first thing that senses that microbe and starts other processes happening. So who is that cell that is hanging out in the tissue and ready to start the next phase of things happening? that cell is going to be a monocyte or a macrophage, at least for our purposes. So we've got monocytes and macrophages that live in all sorts of parts of the body that are going to be ready to sense a microbe if that microbe comes into that part of the body. I call all of these monocytes and macrophages for our purposes. There are unique cells in different organs that are just macrophages, but they got a fancy name from people who do non-immunology things. So if you guys have ever heard of Kupffer cells in the liver, they're macrophages. You ever heard of microglia in the brain? They're macrophages. You ever heard of Langerhans cells in the skin? They're macrophages. So there's a whole bunch of these macrophages that live in different locations of your body and that are trying to sense a microbe when that microbe comes in. And so the first thing that's going to have to happen is that macrophage is going to need to know that there's a microbe present and start to do something. How would, let's imagine some foreign student who's not a member of our class suddenly came into our class. How would I know that there was a foreign student in our class who is not a member of our class? Yeah. Yeah, I would see them and I'd be like, you are not a person I know, right? Like, okay. So do macrophages have eyes? No. So how is the macrophage going to be able to know that there is this foreign cell present? Yeah. Okay, Juliana, what were you going to say? Sorry. So some kind of sensory information, and it might be a molecule on the surface. If we were going to really, really simplify this, what's a word for sensory molecules on the cell surface? Yeah, receptors. So the macrophage is going to have some kind of receptor that's going to help it detect that microbe. This is a really key aspect of our innate immune cells, is that our innate immune cells have a lot of receptors that are really good at responding to things that are foreign and not to things that are self. So they're known as being very good at non-self recognition because they have a lot of receptors for things that are foreign. What I want you to notice about the receptors that are shown here, which I will also show you on the next slide, is that this macrophage doesn't just have one kind of receptor for foreign things. It's got a bunch of them. It's got this triangle one and this square one and this circle one, and it's even got some of these other ones. So it's got a lot of them. So it can recognize a lot of foreign things. Similarly, you can see, this is just some small idea here. And so one macrophage is going to recognize tons of foreign things. It's not like each macrophage is specialized. I say that because when we get to B cells and T cells, it's going to be different. Each B cell and T cell is going to be specialized. But here, one macrophage has receptors for everybody. We tend to call these receptors PRRs. And so PRR, again, hardest part of this, finding chalk. Well, I've already kind of told you it's a receptor, right? That R stands for receptor. Receptors recognize stuff. It's a recognition receptor. And the thing that this receptor recognizes is a pattern that's on microbes. So they're called pattern recognition receptors. And so we're going to have a bunch of pattern recognition receptors on that macrophage so that if a microbe comes around, that macrophage can detect that microbe. Again, both the view on the right and the view on the left are meant to show you that one macrophage will have many different kinds of pattern recognition receptors. So it's not like one macrophage does the mano stuff, and one macrophage does this stuff, and one macrophage does the other stuff. Each macrophage is going to be responding to a lot of different things. And one of the best parts about PRRs is that they recognize some kind of structure that might be common to many microbes. So here you can see we've got 3 different microbes, but they all have the same structure as part of their biology. And the receptor is recognizing just that structure. So you only need one receptor and you get rid of 3 microbes. Usually these structures that are on the microbe that are being recognized tends to be structures that are part of a broad class of microbes. So one famous one responds to LPS, which is in all gram-negative bacteria. It doesn't matter if it's E. coli or Salmonella or Vibrio or whatever. If it's gram-negative, it has LPS, and so we got a receptor that gets all those guys. Similarly, if we're thinking about, there's one receptor that binds to RNA from certain viruses, so it's going to respond to all SARS-CoV-2s and also influenza and also Zika and also long list of viruses that all are RNA viruses, instead of being super specific. And what's nice is also that the microbe can't really mutate to get around these receptors. A microbe can't be like, I don't want to be recognized by that RNA receptor. I'm going to reinvent the wheel and make RNA have a whole different structure. No, it's been evolving for 4 billion years. RNA structure is RNA structure. You're not going to make a new RNA structure to get around this receptor. And so we've got these really conserved structures that are really hard for the microbe to mutate, that are on broad classes of microbes. That's what these receptors are going to be recognizing. The patterns themselves are referred to as MAMPs. This term has actually changed over time. MAMP is sort of the current term, but I will mention that there are, I'll give you an idea of some of the other versions of this. So the MAMP is the part of the microbe that is binding to the receptor in the macrophage. The pattern recognition receptor binds to a pattern. So the P is for pattern here. And the MAMP stands for the microbe-associated molecular pattern. Some examples of MAMPs and their corresponding PRRs are shown on this slide, though I could go on and on and on about different MAMPs and which PRRs bind them, because that's stuff I like to think about. But you can see we've got, again, LPS or flagellin or certain structures of RNA that are in many groups of microbes, and then we have a receptor that it is able to bind. Originally, the term was actually not MAMP. The term was originally PAMP. And instead of saying microbe-associated molecular pattern, people said pathogen-associated molecular pattern. And then we realized these molecules are on good and bad microbes. They're not just on the pathogens. They're not just on the bad guys. They're on the good guys too. So we can't really say these are pathogen-associated molecular patterns because they're on all microbes. So we switched it to MAMPs. But if you see PAMP, it's really the same thing. Later on, we can talk about another related term too, but I don't want to deal with that right now. And there are two things that will happen with that macrophage when the macrophage senses a microbe using one of its PRRs. One of the two things that happens is shown on this slide. Just so you know, the next slide also tries to show you both things. So if you're like, wait, there's the same thing. Yeah, there is going to be a little bit of the sameness between those. So one of the things that will happen is we've got all of these pattern recognition receptors, and that can induce this macrophage to phagocytose the microbe. Remember, we talked about macrophages as being phagocytic cells. If you actually think about the word macrophage, macro big, phage eat. They're big eaters. So the macrophages are going to phagocytose the microbes and degrade them and kill them. So that's one thing that's going to happen when that macrophage finds something using its PRRs. And you can see that same process on the left here. So the macrophage can use its PRR. It can engulf that microbe by phagocytosis, and it can degrade that microbe. So we get rid of that microbe. The other thing that can happen is that we can also see signal transduction happening in the macrophage. So we're going to have signaling, and I'll say more about the signaling other than mystery arrow later. Right now it's mystery arrow. That changes transcription in that macrophage, and now that macrophage makes some new protein products. And so you can see those new protein products being made here. Those protein products are mentioned here as something called inflammatory cytokines, which I will say a little bit more about going forward. But the idea here is that these protein products are, as it says, cytokines. Cytokine is a common word used in immunology that means a protein secreted by an immune cell to try to talk to another immune cell. These are like the messengers of the immune system. This is how one cell tries to talk to another. In some ways you can think about it like hormones. Chemically they're not hormones, but it's the same idea. Like this is the thing one cell secretes to try to tell the other cell what to do. And so when this cell notices a microbe, it starts making messages to send to its friends to help its friends do things differently. These are called inflammatory cytokines because these are the messengers that are involved in inflammation. There's other kinds of cytokines that are involved in other stuff too. Yes, you had a question?
Will both options happen at the same time, or is it just one or the other?
Generally, imagine both options happening at the same time. If we are in a macrophage. If we were not in a macrophage, then it wouldn't. But I'm calling all of this macrophage, and in the macrophage case, both are happening. Yep, David.
So when it comes to like commensal bacteria, how is these kind of responses not going to happen all the time?
That is a great question that many people are researching. So David's question was about, we have microbes that help us. We have good microbes that live in our body all the time. How come we're not doing this all the time without those good microbes? And my answer is, I don't know. A lot of immunologists are doing experiments to try to figure out exactly the answer to that question. Because we have some answers, but we don't have all the answers. Yeah.
Does the binding, the second option, does the binding cause the transcription to occur?
So the binding is going to induce some other proteins to be involved in a signaling cascade. I'm kind of not dealing with them right now. We'll come back and deal with them a little bit later. Okay? Some of the things that we can have happen because of this transcriptional change are the production of inflammatory cytokines, but they actually aren't the only types of proteins that can be made. We can also see some things. Again, we can get our inflammation. We can also do some things that help adaptive immunity work better. We can get some things that help the the macrophage actually kill the microbe better, so we can make better killing molecules. And we can do things that are going to help with tissue remodeling. This is a big sort of place where we think about things like wound healing and wound repair. And so things that are happening because of that PRR signaling will also lead to things like whether or not you get a scar and how the tissue is rebuilt around that wound. That's another area that a lot of immunologists that we don't have as much understanding of as some of the other ones, but it is sort of key. In fact, I personally have been learning a lot more about the fact that it turns out that a lot of aspects of PRR signaling also activate blood clotting. And so it seems like blood clotting is also like part of, and like how would you clot to stop the wound? How would you heal the wound? Is also probably related to those cytokines and that PRR signaling. There are a bunch of weird infectious diseases where clotting is like a weird factor, and in the past, honestly, since COVID, because COVID has a clotting thing, I personally have learned, and the field has learned, that, oh, it's actually related to the PRRs. It's all together. It's not like a separate thing. Usually they increase blood clotting. And so when you turn on PRR signaling, you'll get excess blood clotting. And so sometimes because some of the microbes will actually kind of try to mess with this process, you get really weird blood clotting phenotypes in certain infectious diseases. And in some infectious diseases, people actually die of the blood clotting issues. And we have only, again, really since COVID, been like, oh wait, that's actually the PRRs. Oh, okay. So on Friday, we're going to start with those products, those inflammatory cytokines that the macrophage has made. And we're going to think about what those products do and how that helps us get rid of the microbe. Tomorrow in lab, like I said, make sure that you have your lab coat and make sure you have your protocol as we are getting ready to work with our mice. Definitely, if you want to kind of talk amongst yourselves and pair up in terms of people who are more or less excited, that is 100% okay. I will see you guys tomorrow.